Thank you, Corinne, and thank you for ACN organizing committee for inviting me. So since I'm serving on the committee, I'm sort of self-inviting myself. But what I'm going to talk to you today is that some technology aspect, what we call systems approach, and how we apply that to improve the clinical outcomes of patients. I think Chris did an excellent job of telling us about the nutrition and how the genetics and genomics interact the nutrition. So what I'm going to talk is more technology-oriented aspect. So this is my disclosure. So this is pretty much, my wife worked for a molecular diagnostic company who do lots of genetic testings of cancer patients. And so after today's talk, I'm hoping that you can understand better that the complex nature of cancer is such a heterogeneous disease. I want you to become familiar with the various technologies that to tackle these different levels of complexity. And also, hopefully, you can become more familiar with the systematic scientific wellness testing approaches that we are applying to both wellness and disease patients. So the first concept I want to deliver is that cancer is really a heterogeneous, complex disease. So what do we mean by that? So in this seminal paper by Hanahan and Weinberger a few years ago, so they listed 10 hallmarks of cancers, including sustaining proliferation and resist to cell death, and induce angiogenesis and avoid immune destruction, so on and so forth. And all of these hallmarks can be leveraged to develop new therapies. So one thing that I want to emphasize that there is this intertumoral heterogeneity because each tumors are different. So my tumor or other people's tumors, so they are all different. And so another concept is that they are intratumoral heterogeneities. Even within the same tumors, the cell from cells are different. So I can give you a one oversimplified example is that in this cancer, there are normal cancer cells, but there are also so-called cancer stem cells. So if a therapy specifically eliminates the regular cancer cells, but leave the cancer stem cell alone, the cancer always come back. That's why we are seeing the relapse of all these different kinds of cancer. And those relapse normally what kill the patients. So in developing therapies, we have to target both these heterogeneous tumor cells and also the cancer stem cells, which are the culprit of the relapsing and drug resistant and so on and so forth. So, and another point I want to make is that cancer is systems disease. You know, you cannot just focus on the tumor tissue alone. You have to treat the cancer patient as a whole, as a system, because human body is a complex system. We view this as a network of networks. We have, you know, about 30,000 genes, and all these genes encode mRNAs and proteins, which form molecular networks, and those protein molecular networks form cellular networks and organ networks. Even our human beings, we are social animals because we interact with each other, so through all these social networks. So we call it network biology. And how do we study that? And also, so the nutrition, you know, for persons with cancer, we know that nutrition is so important. That's why we are all here to discuss about this. We know that good nutrition is especially important if one has cancer because both the illness and the treatment can affect cancer. And also, the cancer treatment can also affect our bodies, how we tolerate certain food, and how do we use nutrients. And also, nutrition is, in some cases, is an important part of cancer treatment. So these are, here I list some of the risk factors for colorectal cancers. That's one of the cancers we study. So you can see that many of those risk factors are related to the lifestyle factors, you know, certain food you eat, and whether you exercise regularly, and obesity, smoking, and we use alcohol or not. So on top of that, and there are other risk factors like age, personal family histories of polyps and inflammation, bowel disease, and so on and so forth. But the thing we want to emphasize is that nutrition is really a critical aspect of certain cancer types, especially for the GI system cancers. And in this guideline from American Cancer Society, this is the second edition published a few years ago, that the cancer treatment requires treating the tumor as well as patient as a whole. So how shall we study cancers? So as a system biologist and cancer biologist, so we call it a systems approach to study cancer. So what do we mean by that? So in the past few years, Dr. Leroy Hood, which is the founder of ISB and P4 Medicine Institute, and myself and some others, we have been writing about how we study biology and disease, and including cancers, using systems approaches. And also, we call this a new model of medicine, P4 medicine, predictive, preventive, personalized, and participatory. We think that's going to be the future of healthcare, and I will allude that. I will talk a little bit more later on. So I'm going to introduce a little bit about ISB, System Biology Institute. It's really a driving force in technology development. So if there are existing technologies, we adopt those technologies. If those technologies are not available, we develop new technologies. Then we disseminate those technologies to the broader society to benefit the researchers and hopefully eventually the patient. So at ISB, we do have the state-of-the-art imaging facilities. We have all the gene expression technologies. We have the largest, perhaps the largest proteomics core in the West Coast. And we have a very nice single-cell analysis technologies. So I'm going to introduce a little bit of each and how we apply those to study cancer. So at ISB, we are more or less brainwashed by this idea that biology drives technology, which drives computation data analysis, because during these iterations, there are new insights and new technologies and new software that will be developed. So that can help us better understand biological systems. For disease and for medicine, so what we call P4 medicine, so we believe that clinical need really drives technology development, which in turn drives computation. And at the end of the day, we really want to accomplish better diagnostic and better therapies. So those are really the holy grail of systems medicine and P4 medicine. So this P4 Medicine Institute is just a facilitator of this whole process. The clinical need are the drivers, and the technology and the computations, we think those are enablers in this process. So we believe that eventually everybody will be surrounded by the billions of data points. So we call it a personalized, dense, dynamic data cloud. So we will have your genome, your entire genome, your proteome, epigenome, transcriptome, and your metabolome, so on and so forth. All of this omic data is generated through all these different technologies. And with that, this data can really provide insight into both wellness and disease, and provide the essence for what that precision medicine is all about. So at ISB, we have a very strong family genomic group that led by Gustavo and Jared Roach. So we recently released this largest SNP database with over 150 million single nucleotide variants. And also this group is the first in the world that using whole genome sequencing in a family of 4 to identify rare disease mutations. So that paper was published a few years ago in Science. And in terms of cancer genome, so ISB is also the center for this TCGA Data Analysis Center. So I don't know how many of you are familiar with the Cancer Genome Atlas. So this is NCI and Human Genome Institute. They joint force put together $200 million, and they sequence a few dozen of the cancer samples, including their genome, transcriptome, gene expression, epigenome, so on and so forth. But they have funded 8 data analysis centers, and ISB is one of those centers led by Dr. Ilya Shmulovich. So after the TCGA finished a few years ago, they also funded this project called Cancer Genome Cloud. So to take all this TCGA data into the cloud so that people can do these, you don't have to download the whole thing, but you can do this thing, everything in cloud. So this is a collaboration of ISB with Google and SRI International. And For gene expression analysis and single cell analysis, we really have the state-of-the-art platforms for all sorts of analysis. We have the array-based Affymetrix, the Agilent array technologies. We have next-gen sequencing from Illumina, and we have the Fluidigm Bellmark, which is qPCR-based technologies. And we also have this NanoString encounter called encounter analysis systems. NanoString is a spin-off company from ISB. So ISB is nonprofit, but in the process, in the last 17 years, we have spun off more than 10 companies. NanoString is one of them, which went IPO a few years ago. And in addition to the second generation of a sequencer, we also have available to us this third generation called Nanopore, developed by the Oxford Nanopore. So it's also really small, about your size of your cell phone. So that's a small device that can allow you to sequence a really long piece of DNA. So with all these technologies, how do we apply that to improve the clinical outcomes? So these slides show you the top 10 drugs currently used in the market. So only the orange ones are the patients that this drug will have a positive effect. For the rest of them, the drug does not have any effect at all. So that means we really have to stratify the patient population so that we can identify the population that will benefit from a particular treatment. So how do we apply the system approach to stratify these cancer patients? So I will just give you a few examples how we study cancers with these systems approaches. So the first study we did is a leukemia. So as I said before that, in these cancer populations, there is a special population called cancer stem cells. And you have to target those cancer stem cells to eliminate, to cure the cancers. So what we did in this study a few years ago with Dr. Earl Weissman and Michael Clark, both are the leaders in the cancer stem cell field at Stanford. So we isolate cancer stem cell from AML, acute myeloid leukemia patients. And we compare those cancer stem cells to normal stem cells in your bone marrow. So we want to find what are the real difference between the cancer roots and your normal stem cells, because both can repopulate the population. So we did a transcriptome analysis, and the figure you see here is that the major networks that differential between the cancer stem cells and normal stem cells, and we published a paper on PNAS a few years ago. So this can allow you to better target these cancer stem cells and spare the normal stem cells. And in a second study we did with the Swedish Neuroscience Institute in Seattle on glioblastoma, which is a very nasty tumor. The overall survival is only 14 months for most patients. So we try to identify the personalized drug therapeutic strategies. So what we did is that we take the brain tumors, we make single cell suspensions, and we enrich for cancer stem cells using stem cell enriching media. Then we apply all these genomics, transcriptomics, and single cell analysis strategies to that. So this analysis allows us to identify all the major mutations in these patients, in these primary cell cultures, and also allows us to identify particular different cell populations in this cancer, including the cancer stem cells, so that we can really better targeting each of those subpopulations with different drugs. So our collaborators at Swedish Hospital right now is running a phase 1 trial to see which of the current FDA-approved drugs can target some of these cancer stem cell populations. And the third study is we just got funded recently by DOD, the Translational Team Science Award, a collaboration with a surgeon in the Department of Surgery at the University of Washington. So what we want to do there is we really want to develop an in vitro system that can allow us to do personalized cancer therapy. So in this case, we do this colorectal cancer liver metastasis, CRCLM. So what we did is we take this tumor, we use a tissue punch, we generate this 6 millimeter cylinder of this tissue. Then we cut them into 250 micron slices. Then we culture this 3D culture in the plate. Then we add different drugs. You can do chemotherapy, you can do targeted therapy, and you can do immune therapy as well, because this is the most similar tissues to the tumor to date. Some people use these xenograft mouse model, but that's very expensive, take a long time. But this, we believe right now is the best in vitro model to testing that. Just give you a flavor about what we did is that. So this, what you see here are the 3 patient tumor select cultures, each treated with 2 chemotherapy scheme called FOLFOX or FOLFIRI. So if we have oncologists here, probably you know that these are the 2 major main treatment for colorectal cancer right now. So then we do a 4-day treatment, and at each of those days, we take the samples and we do whole transcriptome RNA sequencing analysis of that. So the green line you see there is the control without any treatment, and the orange one is treated with FOLFOX, and the purple one is treated with FOLFIRI. So you can see that each of these 3 patients, they respond very differently to these 2 different drugs. The first patient, you can see that nicely responds to both drugs, and the second one, basically no response, and the third one is a mixed responding. So we think that with this in vitro system, once we integrate the in vivo data from our oncologists and surgeons, so we can better understand, can we predict the therapeutic effect of these 2 different drugs, because right now basically it's randomly select one or another, not much molecular information. So we hope we can guide the doctors to use this system to better inform them to choose the right treatment for the patient, because timing is everything. If you give them the wrong treatment to start with, I think the chance of a good clinical outcome are very slim. And the fourth project that we did, we are trying to leverage our single cell analysis capacities to stratify therapeutic response. So what I showed before is that we use this bulk tumor tissue, then we use the bulk RNA-seq gene expression profile to predict therapeutic response. But the caveat there is that when you grind up the tumors, all the cells are mixed together, but we know that these cells are all different. You have the cancer stem cell, you have inflammatory cells, you have the matrix cells, so on and so forth. So that didn't tell you which population your therapy are specifically affecting. So this grant that was funded by the NCI Provocative Questions project, started by Varmus, the NCI director, a few years ago, is that we take this to the next level. So we want to take the tumor samples, we dissociate, we make the single cell suspension, then we add the drugs. So then we ask, what specific tumor populations are eliminated by each of these drug treatment? So that way we know, what left are the resistant ones to your therapy. So that's as shown in this diagram here. So this is one of our own data from one patient, CRC patient, with roughly 1,400 individual cells. So what you see here, each dot represents the whole transcriptome of an individual cancer cells. So we can map every gene in our genome or every network into each of these cells. So what we are mapping here is the cancer stem cell. We use a marker called CD133, which is a very well-established cancer stem cell marker. It's also called PROM1. So you can see that maybe 20% of them are light up in the red color. So those are the cancer stem cell populations. And you can also use some other markers to identify your favorite population as well. So what we want to understand is, if we give this patient treatment, so which populations are eliminated and which are left. So those are the ones you should target maybe with your next round of treatment. So that's the more recent data we accomplished. So this is another 1,500 individual cells transcriptome. But what you see here, that different color indicate different treatment. The blue ones are the control without any treatment, whereas the yellow cells are treated with FOLFIRI, and the magenta ones are treated with FOLFOX. So what you can see that this blue population here, they are eliminated by both this treatment. So we can look really deep into what are the molecular networks in these cells, so that it can really tell us what they really are and why they are resistant to these 2 different treatments. So these are the data probably a month old, so we are still analyzing this data. And so what I talked before early on is that we are doing this excruciating targeting on tumor tissues, but we know that that's not enough because tumor interact with the environment, with our human body, with all of our organs. So 3 years ago, Dr. Leroy Hood, which is our founder and president, and ISB started this project called the Pioneer Project. Take 100 individuals to study. All of these are healthy individuals. So we want to study them over a course of long term to see how, if we can monitor their entire body with all this omic data, the genome, proteome, metabolome, blood chemistry, so on and so forth. So can we monitor these healthy people and then give a healthy coach to each individual? Can we improve their health over the course of this dynamic monitoring? So the hope is really to develop a whole series of stories that can we develop actionable opportunities that can improve our health. So this is a project, the RB approval project started in 2014. So with a total of 108 individuals enrolled, and Lee Hood and Nathan Price, who was here last year, who was at the meeting last year, I think they co-lead the project. So these are the assays that are measured in these 108 individuals. We do the whole genome sequencing. We identify SNPs, single nucleotide polymorphism, from millions of SNPs. And we did this clinical chemistry lab test for blood, for urine, for salivas, and over 150 clinical chemistry testings, and your entire metabolites monitoring. So this was done by Metabolon, the company. And also we monitor 400 proteins in your blood. So those are 3 times in a year, every quarterly you monitor those. The genome is once. And also we do this gut microbiome 3 times a year for each individual. And also give each individual a Fitbit to monitor your physical activities, routine, your heartbeat and things like that. And among those, during those 1 year period, so you will have a health coach to work with you. So they will go through the data with you and tell you how do you improve your dietary, your nutrition panels, your exercise, and your movement, things like that. So that's a monthly thing in between of all this round of testings. So what we learned from that study that even though everybody thinks they are healthy individuals, so pretty much 100% of people has been identified having these actionable opportunities to improve their health. So basically the figure on the right shows that about 59% have cardiovascular issues, and almost over 90% people have nutrition abnormalities, deficiency in certain of the nutrition components. Of course, there are also inflammatory diabetes related issues as well. So what about vitamin D? I think that's really a favorite for all the nutritionists. So about 90 out of 108 pioneers, they are low in vitamin D. And we know there are 6 genetic variants from 3 genes affect vitamin D absorption that Chris gave an excellent background already. So, and also there are also other issues affecting vitamin D association. We know that vitamin D can affect various health-related conditions, risks, and also cardiovascular disease, Alzheimer's, and asthma, and even cancer. So what you see here is that it's my own data. So basically, I have 4 measurements of vitamin D from 2015 all the way to 2016. So the initial 2 times is pretty low. I got 20, that's the nanogram per mil. But then my coach helped me to suggest me to take the vitamin D 1,000 units a day, and I did that. So over a period of time, I did go up to 30, and then I got lazy and traveled too much and dropped the vitamin D, so I need to go back to 23 now. So I think I'm waiting for my next test right now, and I try to exercise more, to get more sunshine in Seattle, which is sometimes challenging, but I think this really help with the health coach and all this nutrition counseling is helping all the individuals that accomplish their goal. And another important part is that every individual's got their whole genome sequenced, and we all understand what GWAS means right now from Chris' talk. So what's unique about this program and about ISB is that we have a collection of a few thousand whole genome data. So we can calculate these polygenic risk factors based on those. So let's see, we have the for many of these disease conditions, you have probably dozens, in some cases even hundreds of SNPs can affect those particular conditions. So you can calculate this risk distribution in this overall 2,000 genome that we have in collection. For instance, this LDL, that's the distribution of the population. And if you calculated according to this GWAS data, you can show that whether you are high or low on in this different in this graph. So we can also calculate diabetes risk and also the Alzheimer's risk and so on and so forth because of the available GWAS data for certain ethnic populations. And so also breast cancer risk as well. And these are the list of 60 or so diseases that we can calculate the risk factors based on the GWAS study. And this is one example for LDL cholesterol in the 108 individuals. You can see this is the distribution from very low to very high risk. And when you measure their blood level of LDL cholesterol, so you can see a nice correlation from low to high. In a high-risk patient, you really have high cholesterol levels. And in addition, there are some other metabolites and proteins that also correlated very nicely with cholesterol levels. So what about gut microbiome? So we know that this is another important aspect of our health, and also will be covered by this conference later on. So again, this is my own data. So what I was told that my diversity of gut microbiome is not very good. So that's the average. Most people have a diversity score of 4. And with the what my coach telling me is to eat certain kind of food and more probiotic or prebiotic kind of dietary. So within a half year, I improve a little bit. So I'm still uphill. So I hope to get somewhere near that that end. So hopefully getting my next test, I will see probably next year if I have a chance, I will tell you. Hopefully not somewhere over here. And there are also this correlation between genetics and gut microbiomes. So this is the risk distribution of Crohn's disease from low to very high. And what you see in the circles are the gut microbe of these individuals. You will see that in the high risk patient populations, that these two gut microbiome, Proteobacteria and Microbia are really relatively high compared to the low-risk population. So that's a nice correlation as well in there. So what we learned from these 108 pioneers, they all got their genome sequenced. So, but what they understand that we can really take control of my own health with proper data and coaching. So you have such a big data, and you have these scientists to help you understand this data, and then you have your health coach to help you to improve based on this data. So that's a really powerful combination. And so your genome really does not control your destiny, and it's just tell your potential, and you can improve your health if you do the right things. And also, we are really less well than what we think. So everybody can have some actionable possibilities. We really have to maximize those possibilities to improve our health. And those papers recently in August published on Nature Biotechnology, and those are the cover story of Nature Biotechnology about the study. So, and what we feel that the healthcare is really moving toward this direction. We call it P4 healthcare. So, you want to personalize individuals' their own data and for their own health. And you want to do this early on so that you can predict what's going to happen next. You don't want the disease to happen and treat them. If you can prevent them, I mean, that's probably not only help the patient, but will also help the entire healthcare system in terms of the cost and benefit. And also we want to encourage all the individuals, healthy individuals and diseased individuals to actively participate in this whole thing, because you really want to control your own destiny. So what's the difference between health and contemporary healthcare? We think that the P4 healthcare is proactive, and the contemporary healthcare is more reactive. And is the P4 focusing on individual data and not versus the population data? And we emphasize both wellness and disease in the P4 health model, and in the contemporary healthcare is focusing pretty much over 99% on disease probably. And also we focus on personalized data cloud and versus this average patient populations. And we propose that to use this personalized data cloud for future clinical trial, this one kind of experiment, but not using the average patient population, because you see that all the current drugs on the market, they are not effective for everybody. So you really want to stratify that so that you give the right people the right drug and the right dose at the right time. And so toward that goal, we have recently, about a half year ago in April of this year, a few nonprofit institutions joined force to form this alliance called P4 Healthcare Alliance. So the founding institution for the alliance include the P4 Medicine Institute and the Institute for Systems Biology, the Personalized Lifestyle Medicine Institute, that's the Jeff Bland's hallmark institute, and of course the Board for Certification of Nutrition Specialist, and Swedish Cancer Institute, which is one of the largest local cancer institute in Seattle, and of course the Center for Nutrition Advocacy. So what are main goals for this alliance is, so we have 3 goals. Number 1, we want to train practitioners in this whole field, because there's no standard and no course and no certification for the entire practice of P4 medicine. So we hope to establish a system-based P4 healthcare training program for physicians, for nutritionists, for the exercise physiologists, and so on and so forth, for all these healthcare professionals. And we want to get them certified. I think BCNS are really an expert in this arena to get the people trained and certified and accredited. And really, we want to do advocacy, and I think that's what lots of you have done this morning. So, but we want to do that eventually for the P4 healthcare, and we want to do the same thing. And so the milestones for the P4 Healthcare Alliance is that we want to draft a consensus paper about what P4 healthcare and training and certification are all about. So we want to do this regular seminars from all the major stakeholders who are interested in this transformative P4 health system, and to get their feedbacks to improve our initiatives. And we want to build consensus roadmaps for achieving these goals. Of course, we want to recruit anybody who's interested, any companies, any academic medical centers, nonprofit, and so on and so forth, people who share our vision to join us. I don't know whether Mike is in the audience. We already have some companies that are very interested in joining the alliance already. And of course, we want to implement. We don't just want to talk. We want to really get this into work. So we want to do this accredited P4 healthcare training and certification. And as part of that, we really want to do this neutral genomic and neutral genetic as maybe the first step of the entire P4 training process, because I think those are really critical, and what this the entire College of Nutrition can make a major contribution in the whole P4 healthcare field. So This is my final slide. So after participating in this participation, I hope that you can understand that to enhance the effectiveness of cancer treatment, we need to focus both tumor tissues and to focus on individuals as well. And we really want to incorporate this concept of predictive, preventive, personalized, and participatory P4 medicine into the clinical oncology practice. And with that, I want to thank you, the entire team at ISB, at P4 Medicine Institute, and my clinical collaborators at University of Washington, Fred Hutch, Seattle Cancer Research Institute, and all these fundings from NIH, NCI, DOD, and some foundations for the support of our study. Thank you.